Hello everyone. In this video we are continuing the discussion on transcription but with a little focus on the nucleosome structure, and then we continue with DNA methylation and acetylation. But before we move on to the topic of methylation and acetylation, I am sure that you have seen this slide before. Let's have a quick review on the topics over here. So you know that this is the structure of one nucleosome, and the nucleosome is including proteins, and those proteins are histone proteins, and each nucleosome has a structure of octamer including 8 histone, and then the DNA which is wrapping around the nucleus, which is wrapping around the proteins, so make the structure of such one nucleosome like this. And these nucleosomes are making another structure by becoming more condensed and more complex, that is called the 30 nanometer solenoid. Pause the video and tell me which protein in the nucleosome was responsible or was important to the in making this 30 nanometer solenoid. Yes, I think you answered correctly. That's the H1 histone, which is having the important role for making this structure. That H1 histone also is called a DNA linker because it has the function for making this 30 nanometer solenoid. We will have more explanation and conversation on this matter in the next slide as well. So this 30 nanometer solenoid is extending into a little more wraps, or a little more coiling, and then or condensation, and then it finalize with the structure which is called chromatin. Starting with the DNA and then wrapping around the histone proteins by making the nucleosome, and the nucleosome makes 30 nanometer solenoid, and then a little more condensation until reaching to the structure which is called chromatin. You need to know that the prokaryotic DNA also has the supercoiling, but that supercoil is different completely from the eukaryote, which means that prokaryotic DNA may be associated with proteins, but it is not organized by histone, and is therefore sometimes referred as being naked. If you are interested in knowing more about the wrapping or supercoiling of prokaryote, which is not the same as eukaryote, I have already put a link over here, so you can click on the link and then study more about this topic. So again, as I explained, one structure of a nucleosome is being drawn over here, and that's the core DNA wrapping around the nucleosome, and these are 1, 2, 3, 4, 5, 8 histone proteins, and then the histone H1, which has the function for the structure of the 30 nanometer solenoid, and this is the linker DNA. So the H1 histone binds DNA in such a way to form a structure called the 30 nanometer fiber or solenoid that facilitates further packing. Okay, from here, the conversation and discussion on behalf of methylation and acetylation starts. What does that mean? Methylation is the addition of methyl groups to DNA. So you can see if this is a DNA, and then the methyl group has been added in different parts of the DNA. So first step, what is methylation? Methylation is the addition of a methyl group to the DNA. what is acetylation? Acetylation is the addition of acetyl groups to histone. So here we go. This is the difference. In acetylation, acetyl group will be added to histone protein. As you can see over here, histone has different types of proteins, and this is the acetyl group which has been added to this H1 histone. So this is the H1 histone that has been acetylated. So acetylation is not the same as methylation, because acetylation is happening over the histone protein, not the DNA. In methylation, when the methylation is happening, what is the next step? The next step is that the DNA inhibits transcription. So methylation is the way that the DNA can become silenced. So the processes that inhibit transcription, binding the DNA more tightly to the histone and making it less accessible to transcription factors. And in this case, it is forming a heterochromatin, not the euchromatin. So when the DNA structure is wrapped up like this and is become in a way that not accessible for the transcription, then we call it heterochromatin. But in acetylation, which the acetyl group which will be added to the histone proteins, in this case, acetylation promotes transcription. So one inhibits the transcription and the other one is promoting. Because by acetylation, it makes more accessible. It makes the DNA with the histone a little loose and then provide more accessible region for transcription. So it's the processes that promote transcription in binding the DNA more loosely to the histone, making it more accessible to transcription factors. As you can see, it's a little becoming loose and more open, and these areas are available for transcription after becoming extended. And in this case, this structure is called euchromatin. So we have a heterochromatin. I want to draw like this. If this is a heterochromatin, and this would be euchromatin. And a DNA may have different areas. Oops, I don't know what does that mean. Every time I'm using this, that line comes up. So a heterochromatin can be in different areas, same as euchromatin. So depends on the DNA needs. We have some structure like this as well. So when it is necessary to transcribe, then the DNA will be use the mechanism in which acetylation is happening, more exposure to transcription factors in order to get transcribed, and this is euchromatin. However, this is heterochromatin. NB means nota bene or note well. It's a Latin word. Nota bene or note well, methylation of histones can also occur. This process can both promote and inhibit transcription. I don't want to get you in a very confused area or make you confused about this, but when it comes to methylation, we mainly focus on the methylation on the DNA. However, methylation can happen on histone as well. But we are not opening this discussion over here. So just know that the methylation can happen for histone as well. There is a beautiful discussion about methylation and acetylation, because changes in the environment can affect cell metabolism in a way that the processes such as acetylation and methylation will be affected by the changes of environment or by environmental changes. In this figure, let's start from here. This is an egg, a fertilized egg. A fertilized egg has the sperm and egg from mother and father, and we expect that they are free of epigenetic tags or few epigenetic tags, which has been inherited from their parents, but very few has been over the after receiving from father and mother has been freed or has been removed, those epigenetic tags, and they are ready to have their own changes over the development, because that epigenetic means that they need some cells to be activated, some genes to be activated in order to make different parts, and some genes needs to be inactivated, and that also includes the environmental factors that affect these genes in order to become silenced or activated as well by silencing by methylation and activating by acetylation. So if you look at over here, you can see that the embryo develops many cells with a specialized function due to development, and there are lots of epigenetic tags will include during after development, because the cells are becoming differentiated, so they have their own epigenetic tag. And again, as an adult, the same thing, that they have a specialized function and they have lots of epigenetic tags, which they have already got these epigenetic tags from the environment as they are growing. And reprogramming means that if they have a sperm and egg from mother and father, that's the meaning of reprogramming as well, that during this time all the epigenetic tag will be removed, and it's kind of a cell, a fresh cell, in order to grow and get their own epigenetics features from the environment as they grow. I suggest you make sure that you have watched the previous video, slide, and the video on epigenetic, because it's very clearly it is explaining this definition in this text. Small number of genes, epigenetic tags, make it through this process unchanged, hence get passed from parent to offspring. It's talking about here that there are some epigenetic tags that will be unchanged, no matter if it is reprogrammed and no matter if it is coming from parents, and that's why there are some behaviors and some environmental factors can be inherited from the father and mother, and also some behaviors can be inherited from father and mother to the son, to the daughter, or to the child. Nature of science. Looking for pattern, trends, and discrepancies, there is mounting evidence that the environment can trigger heritable changes in epigenetic factors. This is how is this topic very important, and this because this is related to Unit 3. Also, we will discuss that there as well. There is a very nice topic, very nice link I'm going to pause the video over here and go to that link and go to that website so we can have a look at them. Before I move to that slide, can you read this question and think about and pause the video? What evidence can you find that supports the idea that the environment can trigger heritable changes? There is a nice investigation or research which is called Lick Your Rats. You need to pause the video and read the text by yourself. Okay, did you do it? So I'm not going to read everything here. The experiment is that there are some types of rats that they are licking their childs, their pups, and some of them don't. And apparently, for those rats that they are licking their pups get some epigenetic inheritance from the behavior of their mother, and that's so interesting and very lovely, findings from this research. Those rats that got licked very well by their mom, they are so calm. But those rats that they are not licking their childs or their pups, then pups getting more anxious and they show more anxious behavior while they are growing. This depends on the environment and depends on the situation. It's not that if it's the anxious rat is a good or bad. It depends on the environment. It turns out that if those rats which are in more dangerous situation, those that haven't been licked by their mom, they are more anxious and they are more possible survivable comparing to those who are calm. So this is not exactly the good or bad. It depends on the situation and the environment that they are growing. So everything has the purpose. Isn't it great? You can pause the video over here and read it, and then yes, and then you can pause the video over here and read again. So a different kind of inheritance. Okay, in this slide you need a little self-study. Pause the video and read the text over here in the left side and right side. And then your task is compare the diagrams, not the graph. Diagrams mean these, not the graphs. Then analyze the evidence and deduce conclusions. Okay, did you read? So there are two things that you can compare. First of all, these are the diagrams and graphs are showing the methylation and acetylation. Only methylation. Let's see. The images show the mapping of chromosomal regions with differential DNA methylation in monozygotic identical twins. So it shows the methylation, how there are 4 different chromosomes. This is chromosome 1, chromosome 3, chromosome 12, and chromosome 17. And here is a 3-year-old twin, and here is the 50-year-old twins. So this means that this is the 50-year-old twin number 1, and this is 50-year-old twin number 2. The twins 1, twin 2. And then this is a 3-year-old. So we have a 3-year-old, these twins, and the chromosomes are compared. This is the first thing that you need to understand from this picture. And then the second thing is the methylation. green area shows the hypermethylation. So you can compare this twin with the other one by seeing the level of the green, which one has become more green or less green. So hypermethylation means that the high levels of methylation comparison. So green shows the methylation area. And when you are comparing, you can compare between a 50-year-old twin and a 3-year-old twin. So when you compare both of them compared with the chromosome number 1, you see that while they are growing and from the age from 3-year-old becoming 50-year-old, the amount of the methylation has been increased a lot from in the chromosome number 1. So this area which is yellow, and yellow means the similar levels of methylation in both twins. So yellow means the early amount of the methylation and the original level of the methylation. Yellow has turned into green, so which means that highly amount of methylation has happened from 3-year-old to 50-year-old, and the same here, methylation has been increased. So this area has been the amount of the methylation has been increased, and also as you can see this area the amount of methylation has been increased. And the red is hypomethylation, means the low level of methylation. So when we see the red, means that's the low level of methylation. So in that case, for example, in this case, the methylation is very low, which means that it didn't methylate, the methylation didn't occur, or the low level of methylation has been happening at this area, from 3 to 50-year-old. And also you can compare even between two twins. You will see the differences between a twin, like more red here and less red here. So this means that this twin has more methylated than this one, because red shows the hypo, means less level of methylation. So this kind of comparison you can do, and you can see. Another thing that you can compare is that for chromosome 17 is the least amount of the methylation has happened, almost the same or not if you compare. So this means that this chromosome didn't have many functional in terms of methylation, or it didn't have a very important role in methylation, but this chromosome has a lot of role in methylation. As you can see that the amount of the hypermethylation, which is green, is very high. Many area has become green, but this chromosome, even it is bigger, but still there are less and more red comparing to this one. So you can say that the chromosome number 3 has the highest amount of the methylation, and this chromosome number 17 has the least amount of the methylation. So let's see the result and the analysis. Hypomethylation occurs more away from the end of the chromosomes. Chromosome 3 shows the greatest variation, as I explained. Chromosome 3 shows the greatest variation in methylation at both ages, from 3 to 50, and chromosome 17 changes the least, which is here. So she's here. And the level of both hyper and hypomethylation increase with age. Methylation inhibits transcription. As the organism ages, cells becomes more greatly specialized due to more inhibited and promoted DNA. So that's the fact that the more age, then the more specialized and the more methylation, because methylation means silencing the DNA genes, and they are not going to do, they have already done their function, so they don't need to do it again. So that's why you see the more methylation by growing, by age. Number, sizes, and roles of genes varies between chromosomes. Therefore, it is expected that some chromosomes vary more than others in their degree of methylation and how they are affected by time and environment. So it's not that every chromosome has the same amount for being affected by methylation. Okay, so here we finished the discussion on methylation and acetylation, and I hope you enjoyed and understood what I explained. The last two slides is exactly something which is repetitive, and you can pause the video and read. It's about same thing that we discussed before, transcription and translation. What happens in the transcription and translation is exactly the repetitive things. And the machinery of transcription, the presence of RNA polymerase, which is doing the function, and the pre-mRNA, and then after that the splicing mRNA after so many functions become mRNA as a spliced mRNA. So I just read simply, three main types of RNA are predominantly synthesized: the messenger RNA, which is mRNA, a transcript copy of the gene to encode polypeptide, and then the tRNA, which is responsible, a clover leaf-shaped sequence that carries an amino acid in translation, and in ribosomal RNA, a primary component of ribosomes. So our focus is on mRNA, but if you have a question about the DNA, which is making RNA, how many types of RNA, we definitely have three types of RNA. So the enzyme RNA polymerase that binds to the DNA and recognizes the place that is going to start the transcription. And then RNA polymerase also is multifunctional. It separates the DNA strand and synthesizes a complementary RNA copy from the antisense DNA. And the transcription, which is happening from 5' to 3' direction, and it does this by covalently bonding to ribonucleotide triphosphate that align opposite their exposed complementary partner, and using the energy from the cleavage of the additional phosphate group to join them together. So these are the things that we have already discussed on transcription. Once the RNA sequence has been synthesized, RNA polymerase will detach from the DNA molecules. RNA detaches from the DNA, and the double helix reforms. Transcription occurs in the nucleus where the DNA is, and once made, the mRNA moves to the cytoplasm where translation can occur. And these are the structure of intron and exon. As I already explained, that exon is extremely important, so exon will be kept, and the introns will be spliced by the spliceosome complex, and it will become as a form of mature mRNA. So exon are the coding sections, introns are non-coding, and introns will be removed. And this is spliceosome. The complex, which is shaped to remove the intron part, is called spliceosome. Forms and causes the introns to form loops, which allows the exons to be joined. And the mature mRNA contains all the exons, leaves the nucleus to be translated into polypeptide. And the last slide, which is explaining how this alternative splicing can occur and is very important, and it can create the different types of protein only at the level of the alternative splicing. So it's the same gene, but just by switching the different parts of mRNA during the splicing can create protein A or protein B or protein C, because this area, as you can see, like this exon 1 and then exon 2 and then exon 3 and 4 and 5. So the first one could be completely at the same order from 1 to 5, but it can also create another protein by just removing the exon 3. So this protein is including only 1, 2, 4, 5. And the same, this protein is including only 1, 2, 3, 5. It doesn't have number 4. So this type of splicing can create different types of protein without additional gene, without additional, any other thing, just by during the alternative splicing. And that's the reason that we have like 20,000 around genes for human being, but we have more than 1 million proteins. That's the reason. The splicing process above can happen in different ways to the same gene. Particular exons of the gene may be included within or excluded from the mature mRNA. And multiple proteins produced by a single gene. Each protein produced will vary in its biological function. An example of this is the IgM gene, which produces different immunoglobulin, means antibodies, to fight different pathogens. And that's all. Hope you enjoyed this lesson. Thank you very much.